Okay, hello. Uh, well, in our homeworks, there are some simulations that uses very often the DNA and RNA. So I have to give you some information about the DNA, and with the nucleic acid, I'm starting with the nucleic acid because especially the simulation about the optical tweezers. And also there are some simulations concerning this transcription and translations. I have to give some information about the nucleic acids. So what are nucleic acids? Well, nucleic acids, and in particular DNA and RNA, of course, we'll see later how the information is read on DNA through RNA. They are key macromolecules for the continuity of life. DNA bears the hereditary information that's passed on from parents to children, providing instructions for how and when to make the many proteins needed to build and maintain functioning cells, tissues, and organisms. So how DNA carries this information and how it is put into action by cells and organisms in complex, fascinating, and fairly mind-blowing, and we will explore it more detail in the section on molecular biology. Here we will just take a quick look at nucleic acids from the macromolecule perspective. So nucleic acids, macromolecules made out of units called nucleotides, come in two naturally occurring varieties. One is the deoxyribonucleic acid, DNA, and other is ribonucleic acid, and RNA, in short. So DNA is the genetic material found in living organisms, and all the way from single-cell bacteria to multicellular mammals like you and me. Some viruses use RNA, they don't have DNA as their genetic material, but aren't technically considered to be alive since they cannot reproduce without help from a host. In eukaryotes means that they're the cells that they don't have membranes inside organelles, etc., such as plants and animals. Eukaryotic cells have membranes, and they have nucleuses, and every organelle inside the cell has membranes covered. So DNA is found in the nucleus for eukaryotes, a specialized membrane-bound vault in the cell, as well as in certain other types of organelles, such as mitochondria and the chloroplasts of plants, etc. In prokaryotic cells, they are the cells that they don't have any membrane inside the organelles. In these kind of cells, such as bacteria, the DNA is not enclosed in membranous envelope, although it's located in a specialized cell region called nucleoid. Okay, these are just the locations of the DNA. So in eukaryotes, DNA is typically broken up into a number of very long, linear pieces called chromosomes. Okay. While in prokaryotes, such as bacteria, chromosomes are much smaller and often circular, ring-shaped. A chromosome may contain tens of thousands of genes, each providing instructions on how to make a particular product needed by the cell. Okay? So, how do we get from DNA to RNA and in turn to proteins? Many genes encode protein products, meaning that they specify the sequence of amino acids used to build a particular protein. Before this information, by the way, what is a protein? Protein is a kind of, again, a macromolecule, and that is mostly proteins are used, to build structures in your body, not only tissues and organs, but also, for example, some systems like immune system needs proteins to fight for the disease, and protein production is the most important part in your body. So and the most important job of DNA is just to carry out what kind of proteins are needed for your body, and the amino acids are just the building blocks of the proteins, and every gene inside the DNA contains the information how to get the amino acids, bind them together to form a unique protein. Each protein consists of amino acids. The order of amino acids, there are I think 23 or 21 types of different proteins, but different amino acids, but the order of these amino acids may produce thousands of different types of proteins. So this needs some information, right? This information comes from DNA, and before this information can be used for protein synthesis, an RNA copy or transcribe, this is called the transcription, of the gene must be first made. From DNA first, the cells must produce RNAs, okay? And this type of RNA is called on the first place for the transcription. The type of RNA is called mRNA is produced because it serves as a messenger between DNA and ribosomes, molecular machines that read mRNA sequences and use them to build proteins. The ribosomes are the part of the cells, the organelles, that they read the information from mRNA, and they use this information to build proteins. So the protein factories are ribosomes in your cells. This progression from DNA to RNA to protein to make protein is called the central dogma of molecular biology. So importantly, not all genes encode protein products. For instance, some genes specify ribosomal RNAs, okay, rRNAs, which serves as structural components of ribosomes, or transfer RNAs, they're tRNAs, clover leaf-shaped RNA molecules that bring amino acids to the ribosome for protein synthesis, okay? This is what you see in this picture. Still other RNA molecules, such as tiny microRNAs or miRNAs, act as regulators of other genes, and new types of non-protein coding RNAs are being discovered all the time. So this information is not complete. There are still researchers to discover different types of RNAs. But most of the time, for the time being, that the most important RNA is the messenger RNA that carries this information from DNA to ribosome. What kind of proteins production is needed is carried by this messenger RNA. All right. So what are nucleotides? DNA and RNA are polymers. They are polymers. In the case of DNA, often very long polymer, and they are made up of monomers known as nucleotides. So polymer is kind of a chain, and every polymer chain has its smallest units. It's called monomer, and these monomers are known in DNA as nucleotides. When these monomers combine, the resulting chain is called polynucleotide. That's why we call DNA as deoxyribonucleic acid, etc. So poly means many. Each nucleotide is made up of 3 parts: a nitrogen containing ring structure called nitrogenous base. Okay, what here is, you see, for example, this one, nitrogenous base. Okay. A 5-carbon sugar a 5-carbon sugar, which is called, okay, which is seen over here. For example, deoxyribose sugar is for the DNA, and at least one phosphate group, and you see over here, this is the phosphate group. This is base, this is the sugar, and this is phosphate group. These 3 parts combine together to form the nucleotide. The sugar molecule has the central position in the nucleotide. Over here you see, with the base attached to one of its carbons. Right over here, this is the carbon, and to this carbon, for example, the base is attached, and the phosphate group attached to another, okay, another carbon over, you see over here. To this carbon, the phosphate group is attached. So let's look at each part of a nucleotide in turn. So nitrogenous bases, the nitrogenous bases, of nucleotides are organic carbon-based molecules made up of nitrogen-containing ring, okay, structures. Each nucleotide in DNA contains one of 4 possible nitrogenous bases. Adenine, they are called adenine, guanine, this is adenine, guanine, cytosine, and thymine, and in RNAs, instead of thymines, it is used uracil. Okay, there is a difference in the RNA. In place of thymine, RNA contains uracil. Okay, and they are purines. Okay, adenine and guanine are called purines, and cytosine, thymine, and uracil are called pyrimidines. Okay, purines are meaning that their structures contain 2 fused carbon nitrogen rings, and cytosine and thymine, in contrast, are pyrimidines and have single carbon nitrogen ring. This is the difference. RNA nucleotides may also bear adenine, guanine, and cytosine bases, but instead of thymine, RNA, as I said, they have another pyrimidine base called uracil. Okay, as letter U represents the uracil, as shown in this figure over here above. Each base has a unique structure with its own set of functional groups attached to the ring structure. In molecular biology shorthand, the nitrogenous bases are often just referred to by their 1-letter symbols A, T, G, and C and U. DNA contains A, T, G, and C, while RNA contains A, U, G, C. So in place of T, RNA contains U, which is uracil. Sugars show, in addition to having slightly different sets of bases, DNA and RNA nucleotides also have slightly different sugars. As I said, the 5-carbon sugar in DNA is called deoxyribose, while RNA, the sugar is ribose. So this is the difference between deoxyribose and ribose over here. Okay, so do you see the difference? Well, the difference is right over here. In deoxyribose, this is a hydrogen attached to the carbon, the other again a hydrogen, but in ribose, one of these hydrogen is replaced by a OH. Okay, these two are very similar in structure with just one difference that I just showed, that the second carbon of ribose bears hydroxyl group, while the equivalent carbon of deoxyribose has a hydrogen instead. The carbon atoms of a nucleotide sugar molecule are numbered as 1', 2', 3', and 4', and 5'. Okay, as shown in this figure over here. In a nucleotide, the sugar occupies a central position with the base attached to its 1' carbon. Okay, so we just label these carbons from 1 to 5, and the base in the deoxyribose, the base is attached to 1' carbon. Okay, and the phosphate group is attached to the 5' carbon. Okay, so what about the phosphate? The nucleotides may have single phosphate group or a chain of up to 3 phosphate groups attached to the 5' carbon as shown of the sugar. Some chemistry sources use the term nucleotide only for the single phosphate case, but in a molecular biology, the broader definition is generally accepted. Start superscript 1 and superscript. In a cell, a nucleotide about to be added to the end of a polynucleotide chain will bear a series of 3 phosphate groups. When the nucleotide joins the growing DNA or RNA chain, it loses 2 phosphate groups. So in a chain of DNA or RNA, each nucleotide has just 1 phosphate group. And about the polynucleotide chains, the consequence of the structure of nucleotides is that a polynucleotide chain has directionality. It has direction. That is, it has 2 ends that are different from each other at the 5' end or beginning of the chain. The 5' phosphate group of the first nucleotide in the chain sticks out. At the other end, called the 3' end, the 3' hydroxyl of the last nucleotide added to the chain is exposed. DNA sequences are usually written in the 5' to 3' direction, meaning that the nucleotide at the 5' end comes first and the nucleotide at the 3' end comes last. As new nucleotides are added to strand of DNA or RNA, the strand grows at its 3' end with the 5' phosphate of an incoming nucleotide attaching to the hydroxyl group at the end, at the 3' end of the chain. So this makes the chain with each sugar joined to its neighbors by a set of bonds called phosphodiester linkage. Okay. Alright, so let's come to the properties of DNA. Deoxyribonucleic acid, DNA, the most important biomolecular entity. Chains are typically found in a double helix. What you see over here, these are adenine guanine or the adenine thymine or guanine cytosine base chains. They are connected through these. Okay, and these strands are the backbones that these nucleotides tied to, and they're wheeled around each other to form the double helix, the structure in which 2 matching chains are stuck together, as shown in the diagram. The sugars and phosphates lie on the outside of the helix, as you see over here. Okay, forming the backbone of the DNA, and this portion of the molecule is sometimes called the sugar phosphate backbone. The nitrogenous bases extend into interior, like the steps of a staircase, and in pairs. The bases of a pair are bound to each other by hydrogen bonds. If you are curious about how these are bound to each other, it is because of the hydrogen bonds. This is called a kind of chemical bond. It's called hydrogen bond, and this is between 2 hydrogen atoms. Okay, so the 2 strands of the helix run in opposite directions, meaning that if this is one strand and if this is other strand, you see one strand starts from 5' and ends in 3', but the other strand goes in the opposite direction. It starts from 3' over here, goes in the 5'. Meaning that the 5' end of the one strand is paired up with the 3' end of its matching strand. This is referred to as anti-parallel orientation, and is important for the copying of DNA. This directionality is very important in copying of the DNA. It plays the major role in copying. So, can any 2 bases decide to get together and form a pair in double helix? The answer is definitely no, because of the sizes and the functional groups of the bases. Base pairing is highly specific. A can only be paired with T, and C can only pair with C. Sorry, G can only pair with C, as shown over here. This means that the two strands of a DNA double helix have very predictable relationship to each other. For example, if you know that the sequence of one strand is 5 prime, AA, TT, GG, CC 3 prime, as you see over here, and that means the complementary strand must have the sequence 3 prime, TT, AA, CC, GG, and 5 prime. So this allows each base to match up with its partner. Okay. So when two DNA sequences match in this way, such that they can stick to each other in an anti-parallel fashion and form a helix, they said to be complementary. Okay. So ribonucleic acid RNA, unlike DNA, is usually single-stranded. Okay. A nucleotide in an RNA chain will contain ribose, the 5-carbon sugar, one of the 4 nitrogenous bases, A, U, G, or C, and a phosphate group. Here we'll take a look at 4 major types of RNA: messenger RNA, mRNA in short, ribosomal RNA, rRNA in short, and transfer RNA, tRNA in short, and regulatory RNAs. Messenger RNA, mRNA, is an intermediate between a protein coding gene and its protein product. If a cell needs to make a particular protein, the gene encoding the protein will be turned on, meaning an RNA polymerizing enzyme will come and make an RNA copy or transcript of the gene's DNA sequence. The transcript carries the same information as the DNA sequence of its gene. However, in the RNA molecule, the only difference is the base T is replaced by uracil. For instance, if a DNA coding strand has the sequence 5' AATT GC 3', the sequence of the corresponding RNA will be 5' AAUU GC 3'. Okay. Once an mRNA has been produced, it will associate with a ribosome. Okay, with a ribosome. A molecular machine. It's a molecular machine that specializes in assembling proteins out of amino acids. The ribosome uses the information in the mRNA to make a protein of a specific sequence, reading out the mRNA's nucleotides in groups of 3 called codons. These are called codons. 3, and adding a particular amino acid for each codon. You see these spherical objects. There are amino acids, and you see over here these amino acids are just chained in some order, okay, and they are produced by reading out this information in the mRNA, okay, and they are all done in this unit called ribosome. It's a factory, kind of factory, it's a molecular machine, and we have some simulations that we see how these ribosomes are producing these proteins by reading out this mRNA information. Okay, a ribosomal RNA is a major component. It's a major component, okay, of ribosomes, where it helps mRNA bind in the right spot so its sequence information can be read out. Some rRNAs are also act as enzymes, meaning that they help accelerate or catalyze chemical reactions. In this case, the formation of bonds that link amino acids to form a protein. They act in the process of forming these amino acid chains to make a protein. RNAs that act as enzymes are known as ribosomes, okay, because they're enzyme. Well, what you see over here is a tRNA molecule. It's in three-dimensional structure. Transfer RNAs are also involved in protein synthesis, but their job is to act as carriers to bring amino acids to the ribosome, ensuring that the amino acid is added to the chain is the one specified by mRNA. So they are kind of transferring amino acids to the ribosome. Transfer RNAs consist of a single strand of RNA again, but this strand has complementary segments that stick together to make double-stranded regions. Right? This base pairing creates a complex 3D structure important to the function of the molecule. And regulatory RNAs or miRNAs and siRNAs. So some types of non-coding RNAs that do not encode proteins, help regulate the expression of other genes, such as RNAs may be called regulatory RNAs. For example, microRNAs and small interfering RNAs or sRNAs are small regulatory RNA molecules about 22 nucleotides long. They bind to specific mRNA molecules with partly or fully complementary sequences and reduce their stability or interfere with their translation, providing a way for the cell to decrease or fine-tune levels of these mRNAs. These are just some examples out of many types of non-coding and regulatory RNAs. Scientists are still discovering new varieties of non-coding RNAs. And we will see these regulatory parts in some of our forthcoming simulations. Other kinds of regulatory RNA, such as long non-coding RNA, in short, inc-RNA, and PV interacting piRNA have also been discovered, but their functions are not as well understood. There is growing evidence that miRNAs and other small non-coding RNAs are involved in certain human diseases, including some genetic diseases or cancers. In addition, researchers are developing artificial microRNAs as therapeutic tools to treat human diseases. And nowadays, every day you see this biotech firm, they just discovered this vaccine using mRNAs. In fact, previously they were researching on a type of vaccines that they use mRNAs, microRNAs, to cure or to therapy the disease like cancers. These are just a few examples of regulatory RNAs. There are many others, and more continue to be discovered over time. So this is the summary that just to put almost everything that I said in this presentation. We have DNAs and RNAs, and as the functions of the sugar, the structure, the bases in comparison to each other. You can see this is a table. Okay, so that's it, all about the nucleic acids.